Go ahead and
Okay,
go.
this is group two, and I'm Tiana. We're doing a PowerPoint on the regulation of gene expression. Okay, so information in DNA is a triplet of codes for one amino acid. These are called codons. Amino acids link together to form polypeptides, which are many peptide bonds of amino acids making a protein. Genes code for polypeptides that control things such as the expression of the traits, which is how we look, the function of the cell, or other genes.
A gene is a sequence of nucleotides in DNA encoding one polypeptide chain or one mRNA molecule. Gene expression is regulated at 5 main levels: the genome, the process of transcription, RNA processing and export from the nucleus to the cytoplasm, the process of translation, and post-translation. The process gene expression in the genome can include DNA alterations, chromatin decondensation or condensations, and histone modifications. The process of transcription can be regulated through interactions between regulatory transcription factors and various types of DNA control elements. This means that specific genes are turned on or off in different tissues and in response to changing conditions. RNA processing and nuclear export can contain alternative splicing, regulated export of mRNAs through nuclear pores. The process of translation can be regulated through the modifying the activity of protein synthesis factors, controlling translation and degradation of specific mRNAs through repressors and microRNAs. Post-translation contains reversibly or permanently altering protein structures and functions and controlling protein degradation rates.
DNA and chromatin modification. So, modification of the genome is the first thing we can do is modify DNA to move it around. And DNA sections called transposons can be moved to different chromosomes. We can also modify the DNA to turn parts of the DNA on or off, which can change the histones. Um, some of the some of the chemical processes are DNA methylation and histone modification. Okay, so in this figure, we have DNA methylation, which is where a process of adding a methyl group to DNA, which is also a transcriptional repressor. The compound that is to the DNA binds to transcription activator, which then binds to a nucleosome, causing them to spread, exposing DNA, allowing it to become methylated. Here in the picture, we have the acetyl histone acetyltransferase, which is binding to the activator. Then it binds to the histone, and these histones favor transcription. Once methylated, they don't like transcription anymore and are now inhibited.
Transcription. Transcription is the process of RNA synthesis. It is using DNA as a template to a transfer of information from one nucleic acid to another. The process goes as such. RNA polymerase binds to DNA at a promoter. Initiation requires the binding of transcription factors to both parts of a promoter on the template strand, DNA strand. From there, the RNA chain is elongated, and after that, transcription is terminated. Transcription factors are a protein that is always required for an RNA polymerase molecule to bind to its promoter and initiate RNA synthesis, regardless of the identity of the gene involved. General transcription factors assemble with RNA polymerase at the core promoter. Regulatory transcription factors are ones that selectively bind to a control sequence located outside the core promoter. Generally, regulatory transcription factors bind somewhere along the lines of the proximal control elements on the left side of the figure below. Proximal control elements lie close to the promoter. There are three common types. Proximal control element is upstream or downstream. There are three kinds. There is an enhancer, which is a transcriptional activator. It typically contains several different control elements within it, containing short DNA sequences that serve as a binding site for different regulatory transcription factors and may be identical to the proximal control elements. A silencer is a transcriptional repressor. It inhibits transcription by competing with activators or by binding to the activators to impede them, and it can also bind to a co-activator complex. Insulators are also known as distal control elements. They restrict enhancers and silencers by creating a physical barrier between neighboring DNA regions, thereby preventing enhancer and silencers from inadvertently acting on both groups of genes. This figure is showing how combinatorial control allows different cells to produce large amounts of some proteins, but prevents them from expressing significant amounts of other proteins. Part A is showing how a full set of activating regulatory transcription factors is present, meaning that the gene is expressed at a high level. On the right side, it is showing that there are no regulatory transcription factors available, meaning that the gene is transcribed at a very low level.
My name is Swart. Next slide. So in this process, alternative splicing is one of the main function of this process right here. It's a production of multiple proteins from a single gene encoding. It is determined by the presence of regulatory proteins of a small nucleolar RNA that bind to the splicing enhancer and the splicing silencer. It's an export of messenger RNA through the nuclear pores to the cytoplasm. RNA exhibits defects in capping and splicing that rarely exported. Also in this process, there are different combinations of intron and exon splice junctions and pre-messenger RNA to produce messenger RNAs that differ in exon composition, thereby allowing production of more than one type of polypeptide from the same gene occur in portions when exons are excluded or included, or if there is an inclusion of introns. Well, in this figure right here, you can see different RNAs are exported in different pathways, which may be facilitated by RNA binding proteins that contain a nuclear export sequence. These exports are receptors for these nuclear export sequences and facilitate protein movement out of the nucleus. You can see on this slide right here, this illustration shows another way in which the same gene can produce different transcripts that in turn can lead to different proteins, such as this IgM, which is secreted, or is heavy chain determining which emergent is produced.
Translation. Hello, I'm Ali. Translation includes the process of altering ribosomes or protein synthesis and activity or stability of mRNA. The protein phosphorylation is amino acid residue which is phosphorylated by a protein kinase by, by the addition of a covalently bonded phosphate group. Once it is phosphorylated, it can only be activated or inhibited. Um, Activity and stability of mRNA depends on how it is degraded. If when it's degraded from the 3' end to the 5' end, it shortens the poly A tail at the 3' end to the stability of the mRNA. Um, this this image basically shows the double-stranded RNAs knocking down the expression of the specific genes. And this image basically shows the microRNA silencing translation of mRNAs. Post-translation. Post-translation or is basically a modification of proteiners, functions, and degradation. It includes the processes of reversible structural alterations, permanent alterations, guidance of protein organ by chaperone proteins, targeting of proteins to intra and extracellular locations, um, interaction of proteins with regulatory molecules and ions, degradation. And with the permanent alteration, the protein cleavage is the irreversible removal of a portion of polypeptide chain. And with permanent, with degradation, ubiquitin is a small protein chain consistent of 76 amino acid. Deglans are internal amino particular proteins to be selected for destruction, and microautography are non-selective protein degradations. They can also be harmful to the cell in certain conditions. This image is a system for regulated protein degradation.